Limbic system.
The limbic system is a complex set of brain structures that lies on both sides of the thalamus, right under the cerebrum. It is not a separate system, but a collection of structures from the telencephalon, diencephalon, and mesencephalon. It includes the olfactory bulbs, hippocampus, amygdala, anterior thalamic nuclei, fornix, columns of fornix, mammillary body, septum pellucidum, habenular commissure, cingulate gyrus, Parahippocampal gyrus, limbic cortex, and limbic midbrain areas. The limbic system supports a variety of functions, including emotion, behavior, motivation, long-term memory, and olfaction. It appears to be primarily responsible for emotional life, and it has a great deal to do with the formation of memories. Some neuroscientists, including Joseph Ledoux, have suggested that the concept of a functionally unified limbic system should be abandoned as obsolete because it is grounded mainly in historical concepts of brain anatomy that are no longer accepted as accurate.
Structure.
The limbic system is the set of brain structures that forms the inner border of the cortex. The components of the limbic system located in the cerebral cortex generally have fewer layers than the classical 6-layered neocortex, and are usually classified as allocortex or archocortex. The limbic system is made up of the limbic lobe and other deep-lying structures. The limbic lobe consists of the parahippocampal, cingulate, and subcausal gyrus, hippocampus, and dentate gyrus. The deep-lying structures include the hippocampus, amygdala, mammillary body, habenular, anterior thalamic nuclei, and olfactory bulb. The limbic system has four major components that form two C-shaped structures. The first C-shape is made up of the hippocampus and the fornix, while the cingulate and parahippocampal gyri form the second C-shape. The limbic system includes many structures in the cerebral precortex and subcortex of the brain. The term has been used within psychiatry and neurology, although its exact role and definition have been revised considerably since the term was introduced. The following structures are, or have been considered to be, part of the limbic system. Hippocampus and associated structures. Hippocampus, required for the formation of long-term memories and implicated in maintenance of cognitive maps for navigation. The hippocampus consists of two horns that curve back from the amygdala. It appears to be very important in converting things that are on one's mind at the moment, in short-term memory, into things that one will remember for the long run, long-term memory. If the hippocampus is damaged, a person cannot build new memories and lives instead in a strange world where everything he or she experiences just fades away, even while older memories from the time before the damage are untouched, a condition depicted in the films Memento and 50 First Dates. Amygdala, involved in signaling the cortex of motivationally significant stimuli such as those related to reward and fear in addition to social functions such as mating. Furthermore, the anatomy of amygdala are two almond-shaped masses of neurons on either side of the thalamus at the lower end of the hippocampus. The amygdala stimulate the hippocampus to remember many details surrounding the situation, as well. Fornix is a C-shaped bundle of axon that carries signals from the hippocampus to the mammillary bodies and septal nuclei. Mammillary body, locates at the ends of the anterior arches of the fornix. It is involved with a process of recognition memory. Septal nuclei, located anterior to the interventricular septum, the septal nuclei provide critical interconnections. The septal area isn't related to the sense of smell, but is the pleasure zone in animals. Hippocampus, required for the formation of long-term memories and implicated in maintenance of cognitive maps for navigation. The hippocampus consists of two horns that curve back from the amygdala. It appears to be very important in converting things that are on one's mind at the moment, in short-term memory, into things that one will remember for the long run, long-term memory. If the hippocampus is damaged, a person cannot build new memories and lives instead in a strange world where everything he or she experiences just fades away, even while older memories from the time before the damage are untouched, a condition depicted in the films Memento and 50 First Dates. Amygdala, involved in signaling the cortex of motivationally significant stimuli such as those related to reward and fear in addition to social functions such as mating. Furthermore, the anatomy of amygdalae are two almond-shaped masses of neurons on either side of the thalamus at the lower end of the hippocampus. The amygdalae stimulate the hippocampus to remember many details surrounding the situation, as well. Fornix is a C-shaped bundle of axon that carries signals from the hippocampus to the mammillary bodies and septal nuclei. Mammillary body, locates at the ends of the anterior arches of the fornix. It is involved with a process of recognition memory. Septal nuclei, located anterior to the interventricular septum, the septal nuclei provide critical interconnections. The septal area isn't related to the sense of smell, but is the pleasure zone in animals. Limbic lobe. Parahippocampal gyrus, plays a role in the formation of spatial memory. Cingulate gyrus, autonomic functions regulating heart rate, blood pressure and cognitive and attentional processing. Dientate gyrus, thought to contribute to new memories. Parahippocampal gyrus, plays a role in the formation of spatial memory. Cingulate gyrus, autonomic functions regulating heart rate, blood pressure and cognitive and attentional processing. Dientate gyrus, thought to contribute to new memories. In addition, these structures are sometimes also considered to be part of the limbic system. Entorhinal cortex, important memory and associative components. Piriform cortex, the function of which relates to the olfactory system. Fornicate gyrus, region encompassing the cingulate, hippocampus, and parahippocampal gyrus, nucleus accumbens, involved in reward, pleasure, and addiction. Orbitofrontal cortex, required for decision-making.
Function.
The hypothalamus is a part of the limbic system, which is a group of four brain structures that is the hypothalamus, the amygdala, and the hippocampus. These are involved in motivation, emotion, learning, and memory. The limbic system is where the subcortical structures meet the cerebral cortex. The limbic system operates by influencing the endocrine system and the autonomic nervous system. It is highly interconnected with the nucleus accumbens, the brain's pleasure center, which plays a role in sexual arousal and the high derived from certain recreational drugs. These responses are heavily modulated by dopaminergic projections from the limbic system. In 1954, Olds and Milner found that rats with metal electrodes implanted into their nucleus accumbens, as well as their septal nuclei, repeatedly pressed a lever activating this region, and did so in preference to eating and drinking, eventually dying of exhaustion. The limbic system also includes the basal ganglia. The basal ganglia are a set of subcortical structures that directs intentional movements. The basal ganglia are located near the thalamus and hypothalamus. They receive input from the cerebral cortex, which sends outputs to the motor centers in the brain stem. A part of the basal ganglia called the striatum controls posture and movement. Recent studies indicate that, if there is an inadequate supply of dopamine, the striatum is affected, which can lead to visible behavioral symptoms of Parkinson's. The limbic system is also tightly connected to the frontal cortex. Some scientists contend that this connection is related to the pleasure obtained from solving problems. To cure severe emotional disorders, this connection was sometimes surgically severed, a procedure of psychosurgery, called upfrontal lobotomy. This is actually a misnomer. Patients having undergone this procedure often became passive and lacked all motivation. The limbic system is often classified as a cerebral structure. This structure is closely linked to olfaction, emotions, drives, autonomic regulation, memory, and pathologically to encephalopathy, epilepsy, psychotic symptoms, cognitive defects. The functional relevance of the limbic system has proven to serve many different functions such as affects emotions, memory, sensory processing, time perception, attention, consciousness, instincts, autonomic vegetative control, and actions motor behavior. Some of the disorders associated with the limbic system are epilepsy and schizophrenia.
Hippocampus. Spatial memory.
the hippocampus has been demonstrated to be involved in various processes of cognition. The first and most widely researched area concerns memory, spatial memory in particular. Spatial memory was found to have many sub-regions in the hippocampus, such as the dentate gyrus, DG, in the dorsal hippocampus, the left hippocampus, and the parahippocampal region. The dorsal hippocampus was found to be an important component for the generation of new neurons, called adult-born granules, GC, in adolescence and adulthood. These new neurons contribute to pattern separation in spatial memory, increasing the firing in cell networks, and overall causing stronger memory formations. While the dorsal hippocampus is involved in spatial memory formation, the left hippocampus is a participant in the recall of these spatial memories. Eichenbaum and his team found, when studying the hippocampal lesions in rats, that the left hippocampus is critical for effectively combining the what, when, and where qualities of each experience to compose the retrieved memory. This makes the left hippocampus a key component in the retrieval of spatial memory. However, Spreng found that the left hippocampus is, in fact, a general concentrated region for binding together bits and pieces of memory composed not only by the hippocampus, but also by other areas of the brain to be recalled at a later time. Eichenbaum's research in 2007 also demonstrates that the parahippocampal area of the hippocampus is another specialized region for the retrieval of memories just like the left hippocampus.
Learning.
The hippocampus, over the decades, has also been found to have a huge impact in learning. Colleagues examined the effects of neurogenesis in the hippocampus and its effects on learning. This researcher and his team employed many different types of mental and physical training on their subjects, and found that the hippocampus is highly responsive to these latter tasks. Thus, they discovered an upsurge of new neurons and neural circuits in the hippocampus as a result of the training, causing an overall improvement in the learning of the task. This neurogenesis contributes to the creation of adult-born granules cells, GC, cells also described by Eichenbaum in his own research on neurogenesis and its contributions to learning. The creation of these cells exhibited enhanced excitability in the dentate gyrus, DG, of the dorsal hippocampus, impacting the hippocampus and its contribution to the learning process.
Hippocampus damage.
Damage related to the hippocampal region of the brain has reported vast effects on overall cognitive functioning, particularly memory such as spatial memory. As previously mentioned, spatial memory is a cognitive function greatly intertwined with the hippocampus. While damage to the hippocampus may be a result of a brain injury or other injuries of that sort, researchers particularly investigated the effects that high emotional arousal and certain types of drugs had on the recall ability in this specific memory type. In particular, in a study performed by Parkard, rats were given the task of correctly making their way through a maze. In the first condition, rats were stressed by shock or restraint which caused a high emotional arousal. When completing the maze task, these rats had an impaired effect on their hippocampal-dependent memory when compared to the control group. Then, in a second condition, a group of rats were injected with anxiogenic drugs. Like the former, these results reported similar outcomes, in that hippocampal memory was also impaired. Studies such as these reinforce the impact that the hippocampus has on memory processing, in particular the recall function of spatial memory. Furthermore, impairment to the hippocampus can occur from prolonged exposure to stress hormones such as glucocorticoids, GCs, which target the hippocampus and cause disruption in explicit memory. In an attempt to curtail life-threatening epileptic seizures, 27-year-old Henry Gustave Molaison underwent bilateral removal of almost all of his hippocampus in 1953. Over the course of 50 years he participated in thousands of tests and research projects that provided specific information on exactly what he had lost. Semantic and episodic events faded within minutes, having never reached his long-term memory, yet emotions, unconnected from the details of causation, were often retained. Dr. Suzanne Corkin, who worked with him for 46 years until his death, described the contribution of this tragic experiment in her 2013 book.
Amygdala. Episodic autobiographical memory (EAM) networks.
The amygdala, another integrative part of the limbic system, is also involved in many cognitive processes. Just as in the hippocampus, memory seems to be impacted by processes in the amygdala. However, it is not spatial memory as in the hippocampus, but episodic autobiographical memory, EAM, networks. The amygdala, as researched by Markowicz, was found to be responsible for the encoding, storage, and retrieval of these types of memories. To delve deeper into these types of processes by the amygdala, Markowicz and his team provided extensive evidence through investigations that the amygdala's main function is to charge cues so that mnemonic events of a specific emotional significance can be successfully searched within the appropriate neural nets and reactivated. These cues for emotional events created by the amygdala encompass the EAM networks previously mentioned.
Attentional and emotional processes.
Besides memory, the amygdala also seems to be an important brain region involved in attentional and emotional processes. First, to define attention in cognitive terms, attention is the ability to hone in on some stimuli while ignoring others. Thus, the amygdala seems to be an important structure in this ability. Foremost, however, this structure was historically thought to be linked to fear, allowing the individual to take action to rid that fear in some sort. However, as time has gone by, researchers such as Pesso generalized this concept with help from evidence of EEG recordings, and concluded that the amygdala helps an organism to define a stimulus and therefore respond accordingly. However, when the amygdala was initially thought to be linked to fear, this gave way for research in the amygdala for emotional processes. Kerbeck demonstrated research that the amygdala is involved in emotional processes, in particular the ventral hippocampus. He described the ventral hippocampus as having a role in neurogenesis and the creation of adult-born granule cells, GC. These cells not only were a crucial part of neurogenesis and the strengthening of spatial memory and learning in the hippocampus but also appear to be an essential component in the amygdala. A deficit of these cells, as Pesso, 2009, predicted in his studies, would result in low emotional functioning, leading to high retention rate of mental diseases, such as anxiety disorders.
Social processing.
Social processing is an area of cognition specific to the amygdala. To be specific, the evaluation of faces in social processing is of particular importance. In a study done by Todorov, fMRI tasks were performed with participants to evaluate whether the amygdala was involved in the general evaluation of faces. After the study, Todorov concluded from his fMRI results that the amygdala did indeed play a key role in the general evaluation of faces. However, in a study performed by researchers Kosick and his team, the trait of truthworthiness was particularly examined in the evaluation of faces. They investigated how brain damage to the amygdala played a role in truthworthiness, and found that individuals that suffered damage tend to confuse trust and betrayal, and thus place trust in those having done them wrong. So Kosick demonstrated that the amygdala was involved in evaluating the truthworthiness of an individual. Yet, a man named Roux, along with his colleagues, expanded on the idea of the amygdala in its critique of truthworthiness in others and performed a study in 2009 in which he examined the amygdala in its role of evaluating general first impressions and relating them to real-world outcomes with his study involving first impressions of CEOs. Roux demonstrated that while the amygdala did play a role in the evaluation of truthworthiness, as observed by Kosick in his own research 2 years later in 2011, the amygdala played a generalized role in the overall evaluation of first impression of faces. This latter conclusion, along with Todorov's study on the amygdala's role in general evaluations of faces and Kosick's research on truthworthiness and the amygdala, further solidified evidence that the amygdala plays a role in overall social processing.
Evolution.
Paul D. MacLean, as part of his triune brain theory, hypothesized that the limbic system is older than other parts of the forebrain, and that it developed to manage fight-or-flight circuitry, which is an evolutionary necessity for reptiles as well as humans. MacLean postulated that the human brain has evolved three components, that evolved successively, with more recent components developing at the top front. These components are, respectively: 1. The archipelagium or primitive, reptilian brain, comprising the structures of the brainstem, medulla, pons, cerebellum, mesencephalon, the oldest basal nuclei, the globus pallidus and the olfactory bulbs. 2. The Parahippocampal or intermediate, old mammalian brain, comprising the structures of the limbic system. 3. The neopallium, also known as the superior or rational, new mammalian brain, comprises almost the whole of the hemispheres, made up of a more recent type of cortex, called neocortex, and some subcortical neuronal groups. It corresponds to the brain of the superior mammals, thus including the primates and, as a consequence, the human species. According to MacLean, each of the components, although connected with the others, retained their peculiar types of intelligence, subjectivity, sense of time and space, memory, mobility and other less specific functions. However, while the categorization into structures is reasonable, the recent studies of the limbic system of tetrapods, both living and extinct, have challenged several aspects of this hypothesis, notably the accuracy of the terms reptilian and old mammalian. The common ancestors of reptiles and mammals had a well-developed limbic system in which the basic subdivisions and connections of the amygdala nuclei were established. Further, birds, which evolved from the dinosaurs, which in turn evolved separately but around the same time as the mammals, have a well-developed limbic system. While the anatomic structures of the limbic system are different in birds than in mammals, there are functional equivalents.
Clinical significance.
Damage to the structures of limbic system results in conditions like Alzheimer's disease, anterograde amnesia, retrograde amnesia, and Cliver-Bucy syndrome.
History.
The limbic system is a term that was introduced in 1952 by physician and neuroscientist, Paul D. MacLean. The first evidence that the limbic system was responsible for the cortical representation of emotions was discovered in 1939, by Paul Cliver and Heinrich Bucy. Cliver and Bucy, after much research, demonstrated that the bilateral removal of the temporal lobes in monkeys created an extreme behavioral syndrome. After performing a temporal lobectomy, the monkeys showed a decrease in aggression. The animals revealed a reduced threshold to visual stimuli, and were thus unable to recognize objects that were once familiar. The French physician Paul Broca first called this part of the brain le grand lobe limbique in 1878. He examined the differentiation between deeply recessed cortical tissue and underlying, subcortical nuclei. However, most of its putative role in emotion was developed only in 1937 when the American physician James Papez described his anatomical model of emotion, the Papez circuit. Paul D. MacLean expanded these ideas to include additional structures in a more dispersed limbic system, more on the lines of the system described above. The term was formally introduced by Paul D. MacLean in 1952. Paul D. MacLean developed the intriguing theory of the triune brain to explain its evolution and to try to reconcile rational human behavior with its more primal and violent side. He became interested in the brain's control of emotion and behavior. After initial studies of brain activity in epileptic patients, he turned to cats, monkeys, and other models, using electrodes to stimulate different parts of the brain in conscious animals. Furthermore, he then recorded the animals' responses and, in the 1950s, he began to trace individual behaviors like aggression and sexual arousal to their physiological sources. He analyzed the brain's center of emotions, the limbic system, and described an area that includes structures called the hippocampus and amygdala. Developing observations made by Dr. James W. Papez, he determined that the limbic system had evolved in early mammals to control fight or flight responses and react to both emotionally pleasurable and painful sensations. The concept is now broadly accepted in neuroscience. Additionally, Dr. MacLean said that the idea of the limbic system leads to a recognition that its presence represents the history of the evolution of mammals and their distinctive family way of life. In the 1960s, Dr. MacLean enlarged his theory to address the human brain's overall structure and divided its evolution into three parts, an idea that he termed the triune brain. In addition to identifying the limbic system, he pointed to a more primitive brain called the complex, related to reptiles, which controls basic functions like muscle movement and breathing. The third part, the neocortex, controls speech and reasoning and is the most recent evolutionary arrival. The concept of the limbic system has since been further expanded and developed by Wallnauter, Lennart-Hamer and others.
Etymology.
The term limbic comes from the Latin limbus, for border, or edge, or, particularly in medical terminology, a border of an anatomical component. Paul Broca coined the term based on its physical location in the brain, sandwiched between two functionally different components.
Academic dispute.
There is recent controversy over the use of the term limbic system, with scientists such as Ledoux arguing that the term be considered obsolete and abandoned. Originally, the limbic system was believed to be the emotional center of the brain, with cognition being the business of the neocortex. However, cognition depends on acquisition and retention of memories, in which the hippocampus, a primary limbic structure, is involved. Hippocampus damage causes severe cognitive, memory, deficits. More important, the boundaries of the limbic system have been repeatedly redefined because of advances in neuroscience. Therefore, while it is true that limbic structures are more closely related to emotion, one must think of the brain as an integrated whole.
Additional images.